I'm Daniel Levine, and this is the Bio Report. While existing immunotherapies have changed cancer care, there are several types of cancers where they have limited or no efficacy. Pheast Therapeutics is addressing that by looking to macrophages, part of the innate immune system. These white blood cells gobble up pathogens, cancer cells, and other foreign substances. Certain tumors, though, can evade their attack by expressing checkpoints that serve as "don't eat me" signals. Pheast is developing macrophage checkpoint inhibitors to block these signals and enlist macrophages in the fight against cancer. We spoke to Roy Maute, co-founder and CEO of Pheast, about how tumors evade the innate immune system, the company's experimental macrophage checkpoint inhibitor, and its initial focus on ovarian and triple-negative breast cancer. Roy, thanks for joining us.
Yeah, great to be here. I appreciate the invitation.
We're going to talk about cancer immunotherapies, Pheast, and its focus on macrophages. Let's start with macrophages, part of the innate immune system. What exactly are macrophages, and what role do they play in keeping cancer in check?
Yeah, absolutely. So macrophages are an important part of the innate immune system, and they've got this really compelling name, which is derived from the Greek for big eater. And that's a perfect description of what they do in the body. These are cells that you can look at under the microscope, and they're kind of wiggling around, surrounding and engulfing and destroying or phagocytosing foreign pathogens, host cells, debris. And they play an important role, obviously, in fighting disease, but also, as you said, in cancer. They're generally the most abundant immune cell type in the tumor microenvironment, and sometimes they comprise upwards of 30 or even 50% of all cells in the tumor. So, there's plenty of them there, and under the right conditions, they have the potential to be part of the solution. They have the potential to attack and phagocytose cancer cells. But more often than not, the cancers evolve to protect itself from this kind of attack.
How do cancer cells evade macrophages?
They can do it in a couple of different ways, and one of the most common that we've studied is by overexpressing immune inhibitory signals, which normally protect the healthy cells of the body from its own immune system. But as is so often the case, with these systems, tumor cells can hijack expression of these immune inhibitory signals. So that's true for different parts of the immune system. For macrophages, we often call these "don't eat me" signals, since macrophages act by eating. And it's really common to find several of the well-characterized "don't eat me" signals overexpressed in cancer.
I know listeners will be familiar with checkpoint inhibitors. To date, the approved checkpoint inhibitors have targeted T cells. What's the case for immunotherapies focused on macrophages?
That's a great question. T cell checkpoint inhibitors are really foundational therapies, and the T cells are a really potent part of the adaptive immune system. But they depend on certain initial conditions to work. So, for example, in order to be a good target for T cell attack, a tumor generally needs to have presentation of peptides that look foreign to the immune system. So that would usually require high mutational burden, or other conditions that clearly mark the tumor cells as abnormal. So there's lots of individual patients and lots of kind of broad indications where T cell checkpoint inhibitors exactly find the conditions that are good for them to act, and there are others where they don't. So even in those indications where they work well, there are instances where they work for a while and then lose their potency. So overall, I would say in the cancer landscape, despite the success of T cell checkpoint inhibitors, there's a clear need for additional options. And for us, macrophages are a really natural fit for that empty space. As I mentioned, macrophages are present at very high numbers in most tumor types. And they're there. In some cases, not only are they not part of the solution, they're directly part of the problem, because some of their functions can directly limit the efficacy of other drugs, including T cell checkpoint inhibitors themselves. So macrophages can have immunosuppressive functions that really dampen the ability of other immune cells to get in and do their job in the tumor. So for us, crucially, they don't have the same dependencies as T cells in order to be induced to attack a cancer cell. So for example, they don't really have that same machinery to identify foreign peptides that are derived from cancer-specific mutations. But that is not to say that they, are just running around gobbling up cancer cells on their own. They need help by breaking down the barriers that they see, namely those don't eat me signals that I mentioned. So that's where a drug like Feast's lead antibody, which we call Feast-01, comes in. It targets CD24, which is an important don't eat me signal that has the effect of really suppressing macrophage phagocytosis.
Well, that's interesting that the macrophages can actually inhibit a T cell response. Does engaging the innate immune system to fight cancer by targeting these cells, does that do anything to engage the adaptive immune system as well?
I think so, under the right conditions. So just as you mentioned, macrophages can be in the way of a functional immune response. And I think there's a variety of different therapeutic strategies that, have been tested, are being tested clinically to address that specific angle, helping macrophages, get out of the way, perhaps by depleting them or perhaps by reprogramming them. But there's also another really interesting angle to this, and that is when macrophages are induced to phagocytose, something like a cancer cell, they can actually help to cross-present some of those foreign peptides to T cells. So they can be part of that priming activity to kickstart an adaptive immune response. And I would say that's a phenomenon that's been pretty well characterized preclinically. There's some really nice papers around that, demonstrating that when you induce macrophages to eat, they can help cross-present. And it remains to be demonstrated that we can actually do this effectively in human patients. But as we think about, the possible angles for the benefit of targeting macrophages in cancer, this is definitely a big one. If we could help kickstart that immune response, that's really beneficial in a number of aspects.
Would you expect these therapies to be used as monotherapies, or would there be benefit in using them in combination with other therapies, particularly immunotherapies?
Yeah. So our lead antibody, which targets CD24, it has really strong monotherapy activity in preclinical models. And so we're really encouraged by that. I would say that preclinical monotherapy doesn't always translate to activity in patients in the monotherapeutic setting. And also, as a therapeutic class, I think a lot of immunotherapies, even including the T cell checkpoint inhibitors, which have been really, really successful, often work best in the combination setting. So when we think about the development of Feast-01, we have in mind, some potential biomarker-driven patient populations where we might be in a position to see monotherapy activity. But for us, we expect most likely the registrational level of therapy, the real benefit we could drive for patients would be in combination settings.
Before we talk about your therapy and development, I just wanted to ask you about the origin of Feast, which grew out of the work of the lab of the noted stem cell researcher Irv Weissman. What was the work that led to the creation of Feast?
Yeah, that's right. So our history really started in Irv's lab at Stanford. Feast has 4 co-founders. Irv is one of them. And then the other 3 of us all did our training in Irv's lab at different points in our careers and his lab's history. So, each one of us in different ways studied aspects of macrophage regulation, which Irv is quite famous in the stem cell field and has done a lot of important work on stem cell biology. He's long had an interest in, the functioning of the kind of mature immune system and certainly ways that we can use that for therapeutic benefit. So, over time, he's been one of the pioneers really thinking about how to harness the immune system for cancer therapy, including by targeting macrophage checkpoints. And it was my co-founder, Amira Barkal, who really identified our lead target, CD24, as part of some genetic screening work that she was doing for macrophage regulators in Irv's lab. So, she and Irv worked to kind of unravel how 24 was working, ways that it was similar to previously described macrophage checkpoints and ways that it might be different. They published that work in a really nice paper in Nature in 2019. And the publication of that paper, I think, really kicked off the process of forming Feast. So Amira, as the pioneer of that work, kind of was really interested to see it translated into a company that could generate a therapy against that really exciting target. So we started the company on the basis of that, exciting lead target, and then adapted some of her discovery techniques to form a platform for us to sort of fill out our pipeline.
So you mentioned CD24, which is the target for your initial therapy. What exactly is CD24, and what role does that play in the cancers you're pursuing?
Yeah, CD24 is an immune suppressive receptor that's been studied, over the years. Certainly prior to Irv and Amira's work, I had described functions in B cells, T cells, dendritic cells. But what I think they really added to our understanding of this target is characterizing CD24 as a crucial don't eat me signal. And they were able to pair that with an analysis of, what does this really look like in cancer? And they see that in certain cancers, there's really high expression of CD24. And that seems to be a big part of functionally protecting them from macrophage phagocytosis. So we think it's part of the kind of evolution mechanism that tumors go through in order to evade the immune system. And so, we're really eager to see what happens when we block that in patients.
Given that you're treating cancers in which CD24 is overexpressed, would you expect to use a companion diagnostic to determine if a particular person's cancer would be appropriate for your therapy?
Yeah, we're really interested in that question. And we've done a lot of work preclinically to try to get our hands around this. And I think we're excited to kind of put that into practice in the clinic. So in short, we think there are some cancers where the vast majority of patients express CD24 at high levels. And so for those patient populations, a companion diagnostic is probably not going to be necessary to really explore the efficacy of our drug. But we know that there's several others that have a subset of the patients expressing CD24 at high level. And that often is marked by CD24 amplification at the genetic level as well. And so as we think about, potential companion diagnostics that could enable us to move into some of these other indications where there's lots of patients, but perhaps it's not the entire population, we think that each of those two approaches, both CD24 expression on the tumor cells or the genetic amplification of the CD24 locus, have the potential to be useful as diagnostics. So we think that's a prime opportunity to explore a biomarker companion diagnostic driven development. So we're looking forward to having the chance to test that in the clinic.
My understanding is CD24 has been viewed as difficult to target. What makes it difficult to target, and how have you been able to address that?
Yeah, it's a bit of a weird receptor compared to other typical immune receptors. And that really is based on its structure. So it's a small unstructured peptide, and it's very heavily glycosylated. So about half of the amino acids in the GPI linked peptide are glycosylation sites. So the academic literature suggests that most of its functional interactions, for example, with Siglec-10, which is an inhibitory receptor that binds it, are going to be dependent on that glycosylation. So it's pretty hard to generate antibodies that have a sufficient degree of specificity that you'd want for a therapeutic when you're binding to glycomodifications, since those are often shared across many different proteins in the body. And then on the other hand, for this particular CD24 peptide, there's really not a lot of exposed real estate that's not otherwise covered up by glycosylation. So, we don't want to select an antibody that's going to be occluded from binding to CD24 in many of its different glycostates. And we observe that very frequently for CD24, where we see on different cell types, there's sort of empirical evidence of differential glycosylation from cell to cell. So we sort of knew all of that as we initiated our antibody discovery campaigns, and we knew that we needed to look very broadly. So we tested quite a large variety of different CD24 immunogens. We tested, different immunization schedules and strategies, different mouse hosts. And then I think a lot of our success in finding the drug that we made our lead is just really stringent and extensive screening for different clone families, and I guess a clear thesis for what we were looking for. So for our part, we decided that it was really going to be important to bind and block as many CD24 molecules as we could on a cancer cell in order to get maximal efficacy. And so as opposed to kind of optimizing for, specific binding to a subset of glycoforms, we really optimized for broad binding. And we found that in our experience, that really translates to the highest possible potency for activating macrophage phagocytosis. So it was a bit of an effort, and I think we kind of took our time to find the right drug, but we're glad we did.
Well, what is your lead experimental therapy and how does it work?
Yeah, we call it Pheast 001. And yeah, that's the result of all that screening that I was just talking about. We think it's a best-in-class antibody against CD24. And it's really tuned, as I said, to bind as broadly across as many different glycoforms of CD24 as we could assess. And in our experience, if you just take, for example, commercial reagents against CD24, you'll see very different binding patterns, whether you're looking at a normal cell or a breast cancer cell or an ovarian cancer cell or others. Often the differential glycosylation that's observed between those cell types will mean different binding patterns for those antibodies. And so for us, as we screened, we were saying, we want to find an antibody drug that binds, as many different 24 molecules on as many different cancers as we can. And that is what Pheast 001 represents within our collection of clones that we screened. So at the same time, we've optimized Pheast 001 to try to minimize macrophage-driven toxicity against normal cells, particularly focused on CD24 positive cells in the peripheral blood. Because I think, within the company, we have a lot of experience with macrophage checkpoints, including the clinical stage. And I think our feeling is that those cells are going to be the most sensitive to macrophage activation. So we wanted to minimize the effect of macrophage phagocytosis on those cells. And I think the final Pheast 001 molecule strikes that balance really well of being extremely potent against cancer cells, but not carrying that toxicity over onto normal cells.
What's known about it from the preclinical studies you've done to date?
Well, we've shown that Pheast 001 binds its target CD24 with great affinity and specificity. As I mentioned, a crucial part of that effort for CD24, not always required for every other target that you might go after, is assessing its affinity and specificity for a broad variety of different CD24 glycoforms expressed by different cell types. So we see that it does exactly what we want it to do, not just on recombinant protein, but on cancer cell lines, primary cancer cells, where the CD24 population is really heterogeneous in that glycosylation pattern. So we're really satisfied with that. And what flows from that is that when we put this drug into a co-culture setting with cancer cells and the macrophages, we can really drive very potent phagocytosis of CD24 positive cells in vitro. And that works in the kind of core indications that we talked about earlier that have this really kind of broad high expression of 24. But we've also expanded it now to other cancer types beyond breast, beyond ovarian, including colorectal, lung, pancreatic, and others. So we see that if a cell has 24 expression above a certain threshold, in general, Pheast 001 can drive pretty strong phagocytosis of that cell type. And so we've carried that in vivo as well. So for us, the in vitro experiments are a great start. We really like seeing this kind of effect in mice. And we've been really pleased to see how potent it is in those in vivo settings. So it shrinks or completely eliminates tumors from multiple models in a large variety of different indications. And we have also had the chance to put it really head-to-head with other macrophage checkpoint inhibitors, kind of historical drugs, for example, against the CD47 class, or against other anti-24 antibodies within our kind of screening collection. And we see that Pheast 001 really outperforms those drugs in that kind of head-to-head comparison. So I mentioned, you asked earlier about monotherapy, and I said it works great as a monotherapy in these models, and that's true. We've specifically gone looking for models where its monotherapy efficacy is not as strong as others. And if opposite settings, we can put together combination therapy studies that really showcase its ability to combine with a lot of different therapeutic classes that would represent kind of standard of care drugs in the indications that we're most interested in.
Well, what's the path forward to the clinic, and when might you expect to be in the clinic?
Yeah, we're really excited about this phase of the company's development. So we're in the final stages of preparing our IND for Pheast 001, and we're planning to initiate our first clinical trial in the first half of 2025.
How broad a set of cancers might be implicated in CD24 macrophage checkpoint inhibitors, and how are you prioritizing the indications you'll pursue?
Yeah, it's a great question, and it's really related to what you were asking earlier about a companion diagnostic. So we thought a lot about this. Our intent is to pursue Pheast 001 development really in a data-informed way. And we think the ultimate vision for this drug is that it could be relevant to a lot of different cancers. But one of the great things that we observe about 24 as a target is that the tumor biology gives us a really clear set of signals to generate hypotheses for where to test it. So there's indications like triple negative breast cancer or ovarian cancer, where we see frequent genetic amplification of the CD24 locus. We see high 24 expression. We see a really high differential expression versus the normal tissue of origin. We see an association between 24 expression and clinical outcome. And then, in those indications, I think it's great that there's also a large unmet medical need. So there's a clear spot for this to fit into the treatment landscape. So those types of indications are at the top of our list. We've got a couple that we've really focused on. But as we have kind of studied these potential different biomarker angles, we see that a subset of other indications. So for example, CD24 is genetically amplified in about 20% of colorectal cancer patients and about 25% of lung squamous cell carcinoma patients. That's a small enough percentage of those populations that they may not be the very first ones that we go to test, but they represent really a large population of patients since those are large indications. And so, we're really hopeful that we'll have the opportunity to test those as well, hopefully in a kind of biomarker driven way.
You're not alone in pursuing macrophage checkpoint inhibitors, although other people are pursuing other targets. Are you looking at all beyond CD24?
Yeah, definitely. We've been really fortunate, from the outset to have as part of the company's research a discovery platform that, as I mentioned, is based on the work of one of my co-founders, Amir Barkal, that is a set of genetic screening techniques designed to find novel immune regulators, especially of macrophages and phagocytosis. So we've done a lot of great science using those techniques, found a lot of kind of potentially novel targets for us to go after that would be relevant for oncology or beyond oncology. So I'm excited about work, and yes, it's absolutely the future of Pheast Therapeutics to go beyond CD24.
Pheast raised a total of $76 million to date. It did so at a time that has been challenging for many biotechs to access early stage capital. What was the discussion with investors like, and how much of that success is tied to the pedigree of coming out of Weissman's lab?
Yeah, we've been really fortunate in that respect. I think since those very early conversations, we've had really tremendous support from our investors. I think they were really excited by the team that had come together, both amongst the founders and the early members of the operating team. And they really were into the science as well. So the company's co-founders and early team, certainly including Irv, are really world renowned in their expertise for macrophage immunotherapy. And I think amongst that group also, there was a great track record of successfully building companies and carrying assets into clinical stage development. So, in those conversations, the investors that we talked to were, really interested in the therapeutic space in general and believed in it and were interested to see a success there. And they were really interested in CD24 specifically as we started to tell them what we knew about the target and how we wanted to go about developing it. So they've also been really big believers in that discovery platform that I mentioned and have given us kind of the mandate from day one to pursue that and to make sure that we were, not just moving forward this lead asset, but also developing a robust pipeline. So yeah, I can't say enough good things about our investment partners. They've been really, really great for the company.
How far will existing funding take you?
Like you said, we raised a pretty strong Series A, and I'm happy to say we've been really efficient with our operations so far. The idea of that Series A was to make sure that we could get our lead asset into the clinic, and I think we're well positioned as we begin that process. So current funding will allow us to take our first product, Pheast-01, into the clinic. We'll continue to build our platform and also even begin IND enabling work with the additional programs all in our current funds.
Roy Maute, co-founder and CEO of Pheast Therapeutics. Roy, thanks so much for your time today.
Yeah, it's wonderful to talk to you. Thanks for the opportunity.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.